No, it's not an offence to him to remove the sign. I wish to protect him from the bad things I might say. So, we are in the era of precision medicine. We've come from an era when we have successfully treated cancers, but we have used relatively blunt instruments, which is why I've put these slides together on that very old-fashioned form of writing device. We were not as clever in Europe as the Egyptians, so we didn't invent papyrus. We used a piece of stone called slate. So my slides are all written on slate to indicate that the past is something we need to leave behind. What is the target of chemotherapy? It's our DNA. But it's an incredibly blunt tool. We live in an era when we're trying to design, and we do successfully design, intelligent drugs that very specifically target to reverse specific biological drivers. Every single cell, apart from the red cells, in our patients has DNA. And potentially it is all a target for that toxic combination we throw at our patients. Is this really the way forward? I don't think it is. It's a blunt tool. It's like the firing squad. They need twice as many soldiers with arms to kill 3 people who are completely unarmed. And I feel sometimes when we throw chemotherapy at patients, we have to do that because that's how we get our best effect. But it really is not perhaps the way in an intelligent and rationally designed world we will need to move forward. And I suppose the argument could be, well, that's because the current drugs are rather blunt, they are poorly targeted to subtypes, etc., etc. Well, let's look at the last very good chemotherapy drug that we invented in breast cancer, the taxanes. This is the meta-analysis data based on tens of thousands of women, a total of almost 45,000 women in randomized trials of putting a taxane into an anthracycline-based chemotherapy regimen. And yes, it makes a little bit of difference. Enough of a difference to use in our clinics, but really, why will we do better in the future? There is an enormous unmet need. The taxanes make a difference, but there's a great chunk of patients who are still going to relapse despite taxanes, on top of all our other chemotherapy agents. And has anybody come across an agent which is a chemotherapy agent that looks better than taxanes? If so, I've missed it. So really, are we going to make progress in that direction? I doubt it. Where I think we are going to make progress, and have to a fairly large extent, is by actually trying to understand the cancers that afflict our patients. And one of the big areas of research, which is already showing benefits, is in the DNA, which is, in fact, what the cancer chemotherapy drugs are supposed to deal with. So our target of our current chemotherapy agents, we are now starting to ask proper questions about. So it's unlikely to me that we will answer those questions with more chemotherapy agents. We can look at the germline, where, for example, in patients who have developed a breast cancer because they've inherited a BRCA1 or a BRCA2 gene, and potentially other genes in the future, we, as a community, as scientists, drug company, clinicians, we've developed agents called PARP inhibitors which specifically target the DNA problem that those patients have that gave rise to their breast cancer. We can also, and there's a lot of work going on here, look at the genomics within the cancer itself, the somatic mutations, where we've discovered evidence to back this philosophy that we've worked with for many years, that cancer is a disease of a disordered genome. The mutational status is not fixed. We know that from studies of heterogeneity. If you take multiple samples from a cancer, you do not find exactly the same altered genome. If you take multiple samples over time, from initial diagnosis, persistent disease after neoadjuvant therapy, to recurrence, the genome is changing. So our current drugs are not dealing with that, because that's why patients are relapsing. So I believe we need to make much more concerted effort to understand this varied and heterogeneous genome in order to make real progress, which I think is very unlikely to be with a rather non-specific, broader DNA targeting agent. And we know breast cancer is not one disease. We're not quite sure how many it is, to be fair, but this is a study of 500 patients with 30,000 different genomic alterations. We are making some ground against that with our current chemotherapy agents, but I think we need a whole new look at this to deal with this multitude of genomic problems. Even if you break the breast cancers down into our classic subtypes, basal, HER2, luminal A, luminal B, you will see, and this is a map across the chromosomes from chromosome 1 to 22 and the sex chromosomes, you will see that these cancers are different. I haven't got a pointer here, but if you look at the second peak along for basals, about a quarter of the way along the slide, it's hardly replicated in the other types. In contrast, luminal A and luminal B have a peak looking like a skyscraper towards the right. It's not there in the other two subtypes. And yet at the moment we give chemotherapy to them all that is essentially the same, because we cannot work out a test that says one chemotherapy agent is better than another. It's a very blunt tool. We made some progress, but not where I believe we'll make it in the future. That same paper broke down breast cancer into 10 genomically distinct subtypes, rather more than we use in the clinic. But I suspect with more research, this will become more than 10 subtypes. We really are looking at much more individualization of understanding the genomics of the cancers in our patients. This is data from a study by Matthew Ellis, which is quite interesting because in the top three, and even if you don't really understand what's going on because it's difficult to read the details from the back, you'll see in the middle of these three circus plots there are very few lines. In other words, the fragmentation of the genome is much less in the top three cancers, which are aromatase inhibitor sensitive, compared to the bottom three, where there are more lines, and yet that's where we throw our chemotherapy. It's the same message. We're throwing a blunt instrument at an awful lot of different changes that we haven't really understood. So when we fixed all this understanding, and we've really started to understand the multitude of the genomic changes in our cancer patients, we are moving towards a near individualized selection of therapy. So unless we're going to have one different cytotoxic for every single cancer type, given that they are blunt tools in the first place, I don't think this is how we're going to make progress. Could we combine the new targeted agents that we are developing to reverse these multiple biological genomic changes with chemotherapy, and maybe keep a role for chemotherapy by a kind of general partner, a bit like having a general practitioner, to your specialist new targeted agent? Well, possibly, but in fact it's not proving that easy to combine them. We can with HER2, but as we're beginning to understand, maybe in fact pushing the chemotherapy isn't the most important thing. Pushing the HER2 targeted therapy with doublet therapies, possibly adding in other agents, hasn't yet rendered the role of chemotherapy useless, but it's certainly reducing the importance of chemotherapy there, and actually it's targeting the biological driver that matters. The patients who develop their breast cancer on the background of a BRCA1 or BRCA2 mutation, PARP inhibitors are looking pretty promising, except when you try and combine them with chemotherapy, where the rather broader, blunter effect of the DNA damaging from the chemotherapy is adding to the toxicity. I suspect PARP inhibitors will be things we use more on their own than with cytotoxics. mTOR inhibitors, they've been combined in one study, one of the Bolero studies, with chemotherapy, but actually haven't added very much. So we're not seeing a real potential that the new biologically driven agents will still give a major role to chemotherapy. I think they're going to push the role of chemotherapy down as the biological targets target the underlying and critical biology. It's a little bit like this cartoon. For those who can't see it clearly at the back, you have a number of animals, from a crow to a monkey to a snake to a penguin, an elephant, a fish and a seal, all being asked to sit an exam in front of a tree. And the examiner says, "I must be fair. I'll give you all the same test. Please go and climb the tree." Well, there aren't many of those animals fit to climb the tree. Each animal, to give a fair judge, needs a different test. And I would argue that the future of therapy for our patients is going to be the same. Characterize the kind of cancer and select the right treatment for that cancer rather than throw the blunderbuss of chemotherapy at them all. Thank you.
Thank you, Professor Cameron, and thanks again for being strict time. And now I would like to invite Professor Alvarez Ricardo, who is the Director of Cancer Research in Atlanta, USA.